do you have a sense as to whether the microbiome that you've studied, is that luminal or tissue localized, or have you not investigated that?
We haven't investigated that yet. I mean, you would need sort of labeling procedures like Manuel's. You would need labeling procedures like Manuel's done for H. pylori, you know, incorporating things like RFP or GFP into the bacteria to really see if that specific bacteria is localized, you know, at the pit or deep within gastric glands. But
Interesting.
I encourage everybody to look at where the bacteria are. Like, I would love to see where the bacteria are in Keith's mice, you know, because if Kage is having an effect on the inflammatory response, my bet is that it's in the glands, not on the surface.
Well, so this is for Dr. Peak. Being a little bit of an amateur, I have been reading the literature partly through Robert's mentorship, and there's this budding number of articles that are talking about migration of oral flora into the stomach as a potential. So I'm wondering if you think that your results confirm that, and secondly, do any of those oral flora have alterations of the bile acid that could enable them to release some of the things, the bile acid, more toxic forms that are carcinogenic that you've identified?
yeah, so that's a great question. I do not know the answer as far as the oral flora, but I've collaborated. It's like I stayed at a Holiday Inn last night, right? But I've collaborated with people who do. And Chen Her's group at Columbia, they have really looked at the relationship between oral bacteria and the gastric microbiota, and there is significant overlap between those populations. And that raises the very tantalizing possibility that you could use, you know, a non-invasive test. You could look at saliva, try to predict somebody's risk for gastric cancer later on. As far as bile acids, you know, when we looked at the gastric microbiome in our iron-deficient H. pylori infected mice, they weren't different between the iron normal mice, but the intestinal microbiota were different. And so I'm not sure if the oral bacteria would be representative of what you see in the intestinal microbiome or not. So.
Great talks, super fascinating. From a very simple level, can I ask you, when you have patients with severe atrophic gastritis, you know, we see like this complete or incomplete type IM where the brush borders effaced, the mucosa's thinned out. What happens to the H. pylori if there are no crypts? What happens when it's that atrophic?
So Manuel can address this as well, but you know, in general, there's a reduction in the colonization density of H. pylori when you have those types of lesions. But a group from WashU, Jose Sands and Jason Mills, reported that in both humans and in mice that have SPEM, they can actually see the bacteria binding to those SPEM cells, and that was really not, you know, recognized before. But they used, you know, advanced microscopic techniques to really focus down on these particular areas using co-staining and things, and they found that the bacteria bound to those lesions. So.
Yeah, and I always wonder why they called it atrophy. I mean, atrophy means that things are shrunken, but actually when you look at the replication of those cells, they're in overdrive. So it was one of the earlier papers about this showed that if you stain for Ki67, those glands that are atrophic are actually almost all proliferative. What you're losing is differentiation, because the cells are trying to keep up with the inflammatory damage. And so I wonder if those are even better places for these bacteria to live when you are not allowing things to differentiate properly, and maybe also for other bacteria.
Could you talk to, you know, I think that a lot of us in clinical practice find that people have their, you know, when they get their endoscopic biopsies, they're Helicobacter pylori negative. So do you think that there's there's no correlation? Like, is there actually Helicobacter pylori there? It's just that techniques now are just not able to find it?
There have been some of the earliest studies, that study that I quoted at the beginning of my talk from David Relman, you know, it was a small cohort of patients, but they found that there was, I think, maybe 15 to 20% of individuals that were H. pylori negative by all conventional testing. When they looked by 16S sequencing, they found evidence of H. pylori in those individuals. So I do think that there is a population that has it probably at a subthreshold level that conventional tests are not really detecting. So.
Yeah, and I think an important concept is how many of these people that then are H. pylori positive, again, is this recrudescence or reinfection? And my guess is it's probably recrudescence from these populations that are founders and persisters and are able to come back.